Good day, students. Welcome back. Today we'll be discussing about fatty acid oxidation and how it's how it's regulated. So we'll be talking about regulation of fatty acid oxidation. So we can start by saying that in the liver we have fatty acyl-CoA that is formed, that is in the cytosolic part of the cell, and it has two major pathways that is it can enters into beta oxidation, and this was already explained, that is occurring in the mitochondria, right? So it means the fatty acyl-CoA that is formed at the cytosolic parts of the cell, it can either go into beta oxidation or it can be converted into triacylglycerol and phospholipids by enzymes that are found in the cytosol. So it can either go into oxidation or biosynthesis. So in the pathway taken depends on the rate of transfer of long-chain fatty acyl-CoA into the mitochondria. So the three steps process in the carnitine shuttle is actually a rate limiting for fatty acid oxidation, and is an important point of regulation. So whenever we have fatty acyl carnitine formed, so it already committed that fatty acyl-CoA to oxidation. So we should take note of that. So whenever we have that trans-esterification, when you have the OH of the carnitine attached to the fatty acyl group of the fatty acyl-CoA, and you have carnitine attached to fatty acid to form what? Fatty acyl carnitine, that is already committing the fatty acid to, or fatty acyl-CoA toward oxidation. So when the fatty acyl group have entered the mitochondria, they are committed to oxidation to acetyl-CoA. Now, let's look at malonyl-CoA. The malonyl-CoA is the first intermediate in the cytosolic biosynthesis of a long-chain fatty acid from acetyl-CoA. So when the fatty acid is going to be committed to fatty acid synthesis, malonyl-CoA is the first intermediate that is produced from acetyl-CoA. So now it increases in concentration whenever the animal is well supplied with carbohydrates. Excess glucose that cannot be oxidized or stored as glycogen is now then converted in the cytosol into fatty acids for storage as triacylglycerol. So in this condition, malonyl-CoA is inhibited the carnitine acyl transferase. So malonyl-CoA serve as now inhibitor to the carnitine acyl transferase to ensure that the oxidation of fatty acid is inhibited. Now, if we look at this diagram, it's trying to explain the regulation, the tight regulation of the fatty acid oxidation. So when the diet provides a ready source of carbohydrate as fuel, oxidation of fatty acid is unnecessary and is therefore down-regulated. So two enzymes are key to the coordination of fatty acid metabolism. We have the acyl-CoA carboxylase, ACC, and this is the first enzyme in the synthesis of fatty acid. And we have carnitine acyl transferase 1, which limits the transport of fatty acid into the mitochondrial matrix for oxidation. So when we have ingestion of high carbohydrate meals, it raises the blood glucose level, and thus in one we have it triggers the release of insulin. High blood glucose triggers insulin secretion. So now we have insulin-dependent protein phosphatase phosphorylates acetyl-CoA carboxylase, and activating it to 3, it catalyzes what? In 3 we have acetyl-CoA. Now it catalyzes the formation of malonyl-CoA from what? Acetyl-CoA, which is the first intermediate of the fatty acid synthesis. And we have this malonyl-CoA, now it inhibiting carnitine acyl transferase 1, thereby preventing the fatty acid entry into the mitochondrial matrix. So And when blood glucose levels drop between meals, so we have um, glucagon that is released and now activate the cyclic AMP dependent protein kinase PKA, you remember, which now phosphorylates the it phosphorylates and inactivate the acetyl CoA carboxylase. It phosphorylates and what? Inactivate. You have low blood glucose glucagon is released, cyclic ANP protein kinase, cyclic ANP dependent protein kinase. It now phosphorylates the and inactivates acetyl CoA carboxylase. Now, when that's happened, we have the concentration of malonyl CoA falling, and now the inhibition of fatty acid entry into the mitochondria is relieved. And now at position 7, we have fatty acid enter the mitochondrial matrix, and now at position 8, we have what we call the and now we have mobilization, and it becomes fatty acyl-CoA now becomes the major fuel because glucagon also triggers mobilization of fatty acid in the adipose tissue. You remember when we talk about hormone triggers mobilization of fatty acid. Now, a supply of the fatty acid begins and arriving in the blood. So you have acetyl-CoA now produced, and we have oxidation. So now beta oxidation enzymes can also be regulated. So we have seen malonyl CoA. Now we have now inhibition of biosynthesis as a result of high level of glucose after dietary meal. And also we are going to see now how beta oxidation enzymes are involved in the regulation. Now, two of the enzymes of beta oxidation are also regulated by metabolites that signal energy sufficiency. So, beta oxidation produces energy. So now when there is ample supply of energy, there's no need to waste resources. It's better to use it for other biosynthetic processes. Now, when we have NADH and NAD ratio is when it's high, we have beta hydroxyacyl CoA dehydrogenase being inhibited. So you should take note. Now, this it's actually let's little bit go back. We talk about now when we have in the liver, when we have our fatty acyl-CoA, when it is committed to fatty acid synthesis, when it's in the cytosolic end, it can be committed for both fatty acid synthesis and what we call the oxidation. When we have the carnitine shuttle, when it is activated and it is now enters the mitochondrial matrix via the carnitine shuttle, it is for oxidation. So at the point of malonyl CoA also, it can be regulated. So now for beta oxidation enzymes also at that point but fatty acid oxidation can also be regulated. So we have high level of NADH and NAD ratio. We have beta hydroxyacyl CoA dehydrogenase inhibited. So it's going to be inhibited when this is high. So in addition, the high concentration of acetyl CoA also inhibits thiolase. So we have beta hydroxyacyl CoA dehydrogenase inhibited by high level of NADH-NAD ratio, and also high concentration of acetyl-CoA, it inhibits thiolase. So this is very important and easy to remember. So we have what we call a peroxisomal oxidation. Now, peroxisome also carry out beta oxidation, and the mitochondrial matrix is the major site of the fatty acid oxidation in animal cells, but in certain cells, other compartments also contain enzymes capable of oxidizing fatty acid to acetyl-CoA. So by a pathway similar to, but not identical with that in the mitochondria. So in plant cells, we have the major site of beta oxidation is not mitochondria, but peroxisome. So in plant cells, beta oxidation occurs, but not in mitochondria, but in peroxisomes. Please take note. So in peroxisomes, the membrane enclosed organelles of animal and plant cells, intermediate for peroxisome is a membrane enclosed organelles of animal and plant cells. So the intermediates for beta oxidation of fatty acid are coenzyme A derivatives, and the process consists of 4 steps as in mitochondrial beta oxidation. Now, we have the first step as dehydrogenation, second step addition of water to the resulting double bond. Step 3, we have oxidation of the fatty acyl, oxidation of the hydroxyacyl CoA to ketone body, and also the fourth and the final step, we have thaiolytic cleavage by coenzyme A. So now, this is now a compartment that now divide we have the mitochondrial size here, and this the other size, which is the what do you call it? We have it as peroxisome or gliosome. We can call it gliosisome or peroxisome. Gliosisome or peroxisome. So let's look at what is happening, and in each stage what is actually happening. So in one difference between the peroxisomal and mitochondrial pathway is the chemistry of the first step. So if you look at the first step in peroxisome, flavoprotein acyl-CoA oxidase that introduces the double bond passes electron directly to oxygen producing hydrogen peroxide. So in the first stage, we have this flavoprotein now passing the electron directly to oxygen to produce hydrogen peroxide, and this strong and potentially damaging oxidant, hydrogen peroxide is an oxidant, is immediately cleaved to water and oxygen by catalyst. So now a second important difference between mitochondrial and peroxisomal beta oxidation in mammal is in the specificity for the fatty acyl-CoAs. The peroxisomal system is much more active on very long chain fatty acids such as hexacosanoic acid, 26 carbon fatty acid, saturated fatty acid, and on branch chained fatty acids such as phytanic acid and pristanic acid. So that's another differences. Now, these less common fatty acids are obtained in the diet from dairy products, the fat of ruminant animals, meat, and fish. Their catabolism in the peroxisome involves several auxiliary enzymes unique to this organelle. Now, the third is the NADH that is formed in the second oxidative state cannot be reoxidized, okay, in the peroxisome or glyoxisome. So reducing equivalents are exported to the cytosol, eventually entering the mitochondria. So this NADH that is produced cannot be what? Reoxidized in the peroxisome. So it cannot be reoxidized, but it can be what? Exported, okay, and it will to the cytosol, and eventually it will enter into the mitochondria for oxidation. So now the fourth is the acetyl-CoA produced by the peroxisome, and glyoxisome. Glyoxysomes is also exported. So the acetate from glyoxysomes, that is organelles found only in germinating seeds, it serves as biosynthetic precursor. You remember in our previous earlier lectures, we talked about the fate of acetyl-CoA in higher plants. So acetyl-CoA produced in the mitochondria is further oxidized in the citric acid cycle. Okay. Now, plants, peroxisomes, and glyoxysomes use acetyl-CoA from beta oxidation as a biosynthetic precursor, to buttress our point here. So now, triacyl glycerol as glucose source in seeds. Beta oxidation is one stage in a pathway that converts stored triacyl glycerol to glucose in germinating seed. So we have seed triacyl glycerol acted upon by lipases to form fatty acid, and this fatty acid enter beta oxidation to produce acetyl-CoA, and they enter into glyoxylate cycle and you form oxaloacetate, which undergoes gluconeogenesis to produce glucose, and this glucose can be converted to sucrose, polysaccharides, metabolic intermediates, nucleotide synthesis, energy production, and amino acid synthesis. So now let's look at ketone bodies. And in humans, we said, and most other mammals, acetyl-CoA formed in the liver during oxidation of fatty acid can either enter the citric acid cycle or it undergoes conversion to the ketone bodies. And these ketone bodies include acetone, acetoacetate, and beta hydroxybutyrate. They are exported to other tissues. So this is the structure of acetone, and this is that of acetoacetate or acetoacetic acid, and this is D-beta hydroxybutyrate. So these are the three ketone bodies. Now, acetone is produced in smaller quantities than other ketone bodies. Is exhaled because it's look at it, it's a small molecule volatile. So it can be exhaled out. So acetoacetate and D-beta hydroxybutyrate are transported by the blood to tissues other than the liver. We call them extrahepatic tissues. So tissues other than liver, extrahepatic tissues, where they are converted to acetyl-CoA and oxidized in the citric acid cycle. They provide much of the energy required by tissues such as skeletal and heart muscle and the renal cortex. So they serve as fuel in these extrahepatic tissues. And the brain also, which preferentially uses glucose as fuel, can adapt to the use of acetoacetate or D-beta hydroxybutyrate under starvation condition. You remember when glucose is unavailable, we have explained that in our introductory classes, that brain also uses these ketone bodies as fuel when glucose is not available. So the production and export of ketone bodies from the liver to extrahepatic tissues allow continued oxidation of fatty acid in the liver, and acetyl-CoA is not being oxidized in the citric acid cycle. So the first step in the formation of acetate is occur in the liver and is in the enzymatic condensation of two molecules of acetyl-CoA catalyzed by thiolase. So we have two molecules of acetyl-CoA to form acetoacetyl-CoA. So we have thiolase now that catalyze this reaction. Now, the acetyl-CoA then condenses with acetyl-CoA to form beta-hydroxybeta methyl glutaryl-CoA, HMG-CoA abbreviated, beta-hydroxybeta methyl glutaryl-CoA, HMG-CoA. Now, we have HMG-CoA now synthase, now utilizing water and acetyl-CoA to form the beta-hydroxybeta methyl glutaryl-CoA. Now, after that, we have then the HMG-CoA is now cleaved to free acetoacetate and acetyl-CoA. Now, this is HMG-CoA layers that's now cleave the HMG-CoA to release acetyl-CoA and form acetoacetate. Now, the acetoacetate is reversibly reduced by D-beta hydroxybutyrate dehydrogenase, which is a mitochondrial enzyme to form D-beta hydroxybutyrate. So we have acetoacetate. We can have acetoacetate decarboxylase to remove carbon from acetone, and also it can be converted to beta hydroxybutyrate by the action of beta hydroxybutyrate dehydrogenase, which uses NADH plus H to give us the NADH plus. So in healthy people, acetone is formed in very small amounts from acetoacetate, which is easily decarboxylated, either spontaneously or by the action of acetoacetate decarboxylase. Okay. Now, because individuals with untreated diabetes produce large quantities of acetoacetate, their blood contains significant amount of acetone, which is toxic. Acetone is volatile and imparts a characteristic odor to the breath, which is sometimes useful in diagnosing diabetes. So in the extrahepatic tissues, D-beta hydroxybutyrate is oxidized to acetoacetate by D-beta hydroxybutyrate dehydrogenase, and this is the enzyme, and this substrate to form acetoacetate. And this acetoacetate is now acted upon by beta-ketoacyl-CoA transferase, which uses succinyl-CoA to donate the acetyl-CoA to donate the coenzyme A group to form succinate and acetoacetyl-CoA. Now, this acetyl-CoA is acted upon by thiolase in the presence of coenzyme A to produce two molecules of acetyl-CoA. Now, this is ready for energy generation. Now, ketone bodies are overproduced in diabetes and during starvation. So in starvation and untreated diabetes mellitus, it leads to the overproduction of ketone bodies with several associated medical problems. During starvation, gluconeogenesis depletes citric acid cycle intermediates, diverting acetyl-CoA to ketone body production. Now, in untreated diabetics, when the insulin level is insufficient, extrahepatic tissues cannot take up glucose efficiently from the blood, either for fuel or for conversion to fat. So under these conditions, levels of malonyl-CoA fall, Now, inhibition of carnitine acyl transferase 1 is relieved. The fatty acid enters mitochondria to be degraded to acetyl-CoA, which cannot pass through the citric acid cycle because cycle intermediate have been drawn off for use in the substrate as in gluconeogenesis. So the TCA cycle cannot degrade acetyl-CoA because this intermediate in that cycle has been removed to be used as substrate for the synthesis of glucose in gluconeogenesis. Okay, production of carbohydrates from non-carbohydrate intermediate. So here we have lipid droplets in this is hepatocytes, this is liver cells. So we have lipid droplets here in yellow, and we have acetoacetate, D-beta hydroxybutyrate, and acetone. They are actually been exported out. So we have acetate and beta hydroxybutyrate. They are exported as energy source for heart, skeletal muscles, kidney, and also brain. But in the okay, in the hepatocytes, that's in the liver cells, we have citric acid cycle, right? Taking acetyl-CoA to produce oxaloacetate, and this oxaloacetate is now taken to produce glucose via gluconeogenesis, and now this glucose is now exported as fuel for brain and other tissues. Okay, do you remember where is the source of the acetyl-CoA? We have what? Fatty acid oxidation, then to form the acetyl-CoA, which can go into ketone body formation, or it can enter into citric acid cycle to form oxaloacetate. Now this oxaloacetate goes to gluconeogenesis to produce glucose, to export it to extra to brain and other tissues, and also the ketone bodies form here also to heart, skeletal muscle, and the brain for energy. So the resulting accumulation of acetyl-CoA accelerates the formation of ketone bodies beyond the capacity of extra hepatic tissues to oxidize them. So the increased levels of acetoacetate and D-beta hydroxybutyrate lower the blood pH, causing the condition known as acidosis. So increased levels of acetoacetate and D-beta hydroxybutyrate lower blood pH to cause acidosis. Now, extreme acidosis can lead to coma and in some cases, death. So ketone bodies in the blood and urine of untreated diabetics can reach extraordinary levels, a blood concentration of 90 milligram per 100 mils compared with the normal level of 3 milligram per 100 mil, and urinary excretion of 5,000 milligram per 24 hour compared with the normal rate of 125 milligram per 24 hour. This condition is called ketosis, when you have ketone bodies in blood and urine, leading to this as high as these levels. So we have ketosis. Okay, thank you for listening and watching. This brings us to the end of the lecture. So we see you in the next class.